A couple of words about me. So I'm a professor of neurosciences at the Sorbonne University in Paris, and I work in an institute which focuses on capacities like learning, memory, perception, the brain, how these capacities are actually mediated by the brain and so on. But my team in particular works, as you can see, with bees. So one of the typical questions I get is that if you're interested in capacities, important capacities like learning, memory, and so on, why do you care about bees? Why don't you go and study that in primates, in monkeys, or things like that, which are closer to us? Indeed, you could have this kind of perspective, for instance, by looking at these two brains. This is the human brain, our brain, and you see it has an impressive volume, okay? And it has, even more impressive amount of neurons. You have 100 billions of neurons in average there, which is impressive, actually, again. And at the same time, you can see here a honeybee brain. Of course, this is not at the same scale, as you can realize by looking at the numbers. This is a real brain, by the way. This is not a scheme. This is a 3D reconstruction of the bee brain using some microscopy techniques we have. And you see, this is a tiny brain, just 1 cubic millimeter, you can see here. And you have 1 million neurons, which is nothing compared to the 100 billions we have. So by looking at these numbers, you might be tempted to think, what is interesting there? Why caring about these little brains and their capacities? Well, one of the answers I like to provide is that these little creatures, actually the honeybee foragers, are actually there are species that have been tuned to actually depend exclusively on learning and memory. So it's a gold mine in a way, because all they do depends on these capacities all day long. We're speaking about honeybee foragers, and these honeybee foragers actually have something that was already discovered and observed by Aristotle long ago, which is something called flower constancy. You probably heard about that, which is the fact that a honeybee forager in a foraging trip does not switch from one flower species to the other. It remains foraging on the same species as long as it provides the reward it's looking for, so nectar or pollen. And this flower constancy actually depends on learning and memory. Why are they constant on a species? It's because actually they have learned and memorized the sensory information that allows them to go back to that flower species. What kind of sensory information? Well, they learn colors, they learn odors, they learn also shapes, they learn also the position in space, which allows them to go back and forth to the same place to exploit given flower species and so on. And that's the basis for flower constancy, as you can see here. So then you have a little creature performing thousands of foraging flights all day long, and all of this is driven by having learned which species and keeping in memory which species and where to go. So for the researcher interested in these capacities, it's like, as I say, a gold mine. You have an organism which depends exclusively on that. The other thing, or the other argument for using bees, is that they are experimentally accessible. What do I mean by that? Is that you can train them and test them in the laboratory. You will see it's possible to train bees to, like you would train a dog or something like that, to fly from the hive into the lab, to enter into specific setups, mazes or whatever you have conceived, to ask specific questions. How good do you learn this particular situation or stimuli? How do you remember? Can you make the difference between these two, and how long does your memory last? All these questions can be asked actually, because as long as you will be providing in your setup a little drop of sugar water, the bee will be coming all day long to work with you. Of course, I say to students, forget the break at noon, because if you go to a restaurant, the bee will be coming, will find nothing, and then your experiment is lost. You are the slave of the bee. Okay, so that's the way it works, actually, because they are cooperative as long as you provide the sugar reward they're looking for. So it's possible to do lots of experiments with them, and then you will have your answers. The other point is where the apparent disadvantage actually becomes an advantage. We were speaking about tiny brain, few neurons, and so on. But this brain is accessible. You will see that we have developed a series of tools and techniques to go into this little brain and see how it works. And then, of course, if you would be looking for circuits or areas responsible for this and that, and I remind you that I'm a neuroscientist, so this is what I would like to know. My question is not, can bees just do this and that? Of course, that's the question, but how? What are the mechanisms that allow this capacity? That I would like to know, and this is why I need to go somehow into the brain. And then the question is, or the point is, it's easier perhaps to find the circuits, the areas responsible for this or that, where you have only 1 million of neurons compared to the 100 billions of neurons we have. So this is what I say. I mean, the disadvantage becomes an advantage, actually. So these are some of the reasons, and the main reason also, which is interesting for me, is that these animals do not have only elemental forms of learning. By elemental forms of learning, I mean something that all living creatures, I mean animals, have, which is learning very simple associations, worms, everything. I mean, all animals can do this kind of learning, like learning, for instance, blue means reward and yellow means punishment. You know, it's a simple association between A and B or C and D. These, of course, these animals will have, like most of animals, but they have something more, something called higher order forms of learning, and this is what makes them interesting. And you will see examples of those. Because some of these forms of learning were considered until recently only to be a prerogative of humans and primates. And yet suddenly we discovered that they are there. And you will see some examples. So, what are the questions for today? So the field I represent is called neuroethology because, of course, we are interested in the behavior of the animals, ethology somehow, but we also go in, or we like to understand the neural substrates of this behavior. And I was telling you that in natural conditions, bees learn and memorize different kinds of information. And the question would be actually, as I say, do we have just simple forms of learning? Somehow I anticipated the answer. The answer is no. But actually, which kind of complex, non-elementary forms of learning do we have in this little brain? Typically, if you see these forms of learning in humans, you will see, oh, this is cognition, like higher order cognitive capacities. Do we have something like these too? And for instance, can bees learn concepts, conceptual relationships? And I will explain that later. Can they count? Could you imagine that bees can count? And if they count, let's imagine that they can count. How do they represent numbers in space? We have a way to represent numbers. We order numbers from left to right. 1, 2, 3, 4, 5, small, large, left to right. Well, who knows? How do they do that? So these are some of the questions I'd like to address today. How good are they at memorizing? Does it take time for them to learn? Is the brain plastic? So by plastic, I mean that our brain changes all the time. New connections are made and so on to stabilize information and so on. Is the same in the brain. Is it a plastic brain? Does it change with experience? And the last question, which we started addressing only recently, or well, not so recently, but nevertheless in the last times, being concerned by what all is happening around us, is how do pesticides affect the brain of bees? That would be the last question I'd like to address today. So the first point will be to show you some examples of higher order forms of learning in bees, to show you that these little creatures are more than what you believe they are. So, and I would like to use for this visual learning, so these will be examples of bees trained to visual stimuli, and using free-flying bees, bees that have been identified with a spot on the thorax to recognize them, and which are flying back and forth between the hive and the lab to solve problems. So, and we will be speaking about conceptual learning, learning concepts. And I'd like to show this very, very famous American psychologist, Thomas Sentel, because he was the one defining conceptual learning, and he used to say is the capacity to learn to solve problems based on relationships between objects or events in the environment. What does it mean? It means that we might solve problems by using relationships, not focusing on the quality of the objects themselves, but on the relationship binding the objects. For instance, you might learn to solve a problem realizing that what gives the solution to the problem is the fact that you have to choose always the larger object. So you have two objects, and one choice is correct, the other is incorrect, and then you realize, okay, the objects are changing all the time, so I cannot focus on their shape, their color, or whatever. The objects themselves, it's not providing or are not providing the cue to the problem, but I realize suddenly, oh, there is a relationship. One is larger than the other, and if I choose the larger one, then I get the solution to the problem. You understand what I mean? So it's not the object, it's the relationship binding the object, which means somehow you have to get rid of focusing on the object and place yourself on a superior level, the level of the relationship binding the object. And then you might have lots of relationships that you might use to solve problems, like choosing always the object that is different from what has been shown to you, choose the larger object, the object that is above a certain references, the object that is to the left or to the right. So again and again, we learn to use this kind of conceptual learning. So the question is, can bees do that? And that's a complicated question, because just to tell you how it works, this kind of conceptual learning is typically used by psychologists working with children to ask the question, from which age a child can do this? From which age? Because it's typically around 5 or 6 years old, they can start finding the solution using these relationships, but before that, they just look at the objects and then they are not able to extract this relationship. So, well, okay, so what about our bees? So this is a question I asked some years ago, and I wanted to know if bees would be able to learn a relational concept, the same we were talking about. And I focus on the concept of sameness. The concept of sameness is very well known, and there is a protocol to study that, which is called the delay matching to sample problem. And the reason why it's called like that, because the experiment goes typically like that. So imagine Roger is my experimental subject sitting there, and I say, Roger, look at this object. You see it? That's my clicker, okay? Okay, you just see it. And now, Roger, I will show you two objects. This one and this one. That's my USB key. And then you have to choose one. And of course, Roger has no idea which one is the right one, but there is a choice that is correct, and there is a choice that is wrong. So, Roger, choose one. The USB key, wrong. Wrong choice. Okay? You, of course, also have no idea what's going on, but there is something. And then I would continue, so I might need another object, but I don't have. I take these. So now, Roger, let's see. You see this object? Okay, now there is again a situation where you have to choose. Now, choose between these two. Now that's correct. Do you know what is correct? You know why? There is a rule. There was a rule which made your first choice incorrect, and now this choice correct. There is a rule, but you have to realize that. This is why it's complex, because people don't realize until get different examples. The rule was simply choose what has been shown to you before, irrespectively of what has been shown to you. So first time I showed to Roger the clicker, and he chose the USB key. It was wrong. Being confronted with the clicker in the USB key, he had to choose the clicker because that has been shown to him. When I show him first the USB, then he has to go for the USB. I can change the objects all the time until you realize, okay, the rule is choose the same that has been shown to you before. And you realize why these problems are complex, okay? So this is exactly the question we ask to the bees. Can bees do this kind of problem solving? And you can see, for instance, here, this is a bee that has been trained to fly into the lab, and coming to this image, there is a little tip in the middle providing a drop of sucrose solution. And if you look well, I don't know how well can you see there from there, but there is a spot, a green spot on the abdomen. So we know this is our bee, the bee we are following all day long, and we use just this bee to do these experiments. And of course, the experiments are done with many bees, one after the other. So, and this is what we did. So we trained the bees to fly into the lab, to go into a Y maze. So they arrive here, and at the entrance of the maze, there is a yellow disc with a hole in the middle. So, and then the bees have to go through the hole and then make a choice. It's exactly like the situation with Roger. So they see something, and then they have to go in. Because if it is the sameness rule, if they go to yellow, they land and there will be a reward. Choose the same, what has been shown to you at the entrance. If they go to blue, it's wrong. They are punished. They are expelled from the maze. They have to do everything again. They hate that. They lose time, and so on. 5 minutes later, they come back to the lab, and surprise, the stimulus at the entrance has been changed. Now it's blue. And blue that was previously punished. But now it's not important. Because blue is shown at the entrance, you have to go for blue. And then the animals should go for blue in this case. This experiment, of course, you take care of having the reward on the left and the right, the same proportions to avoid them turning always to the same side and things like that. Okay, imagine that they learn that when there is yellow, they go for yellow, and when there is blue, they go for blue. Well, that's not enough. You should put or place the bees in a fully new situation to see if they are really using a rule, if they have extracted the rule. And this is what we offer to them. Once they were actually working with yellow, blue, and so on, then they arrive at some point and they saw these. No colors, vertical, black and white, or horizontal black and white gradings. So there are two possibilities. Either the bees are totally disoriented because they see this for the first time and they don't know what to do, or they have a rule. And in this case, they would not care by seeing these for the first time. They would enter and go to vertical, and in this case, they would enter and go to horizontal. So the experiment is done with two groups of animals in both directions. What does it mean? We had a group trained on the colors, yellow, yellow, blue, blue, and so on, and then tested on the patterns, okay? And another group trained on the patterns and tested on the colors, both directions, just to check that the performance was similar. So, and here's the result. So this is the training, okay? So you have two curves, one group trained with colors, so blue, blue, yellow, yellow, and so on, and one group trained with patterns, vertical, vertical, horizontal, horizontal. These are blocks of 10 visits to the maze. So 60 times the bee flying back and forth between the hive and the lab. So to give you an idea, it could be one entire day of work for a bee, okay? More or less. And you see that at the end of the day, both groups are quite good, actually. It's 75% correct choices. This is not bad for a little bee, actually. They are doing pretty well in their respective problems. But now comes the critical moment. When they reach this point, we presented to each group the new situation. So when bees trained with colors here in red reach this point, they arrive and we show them the gratings, okay? And when the sample at the entrance was vertical, already in the first choice, they went for vertical. And if it was horizontal, they went for horizontal. The bees trained with the gratings that you can see here arrived and saw for the first time a blue disc at the entrance, and when it was blue, they went for blue, and it was yellow. If it was yellow, they went for yellow. So this experiment was very exciting to us because it showed for the first time that these little animals do not learn just simple associations. They are able of conceptual forms of learning, and now you understand what I meant by higher order forms of learning when I spoke about that at the very beginning. So here we are far away from simple forms of learning. We are in a case of extracting concepts, abstract relationships, in this case the concept of sameness, of the choose the same, which is applied to different modalities. I don't have time to tell you about these other experiments, but in this work we even show transfer of the rule between colors and others. So it's not just visual, so it applies to different modalities. And also no time for that, but they can learn the opposite concept. So choose always the contrary to what I'm showing you. And they have no problems in mastering that rule too. We show that too. Meanwhile, our team has done many, many experiments on these forms of conceptual learning, and they might learn many, many different concepts, spatial concepts like above, off, below of, to the right of, to the left of, and so on. Or like in this experiment, which was done by one of my PhD students, where they combined two rules at the same time. They learn that they need to have two images, one on top of each other. You see, one and the other. The reward is here in the middle. And on top of that, not only the images have to keep this spatial relationship. If it's like that, it's not good. It has to be vertically arranged. And on top of that, they had to be different from each other. If you place the same image in the appropriate relationship, they don't like it because it's not what they learn. They learn the two images have to be different and in the right relationship. And again, you prove that by changing the images all the time, but keeping the relationships constant, and that worked. So the other thing that is interesting, which is the transition to my next point, is that they are also able to learn numerosity concept. By this, I mean a sense of number. So a sense of number means that they can count somehow. That would be the question. And these were experiments that were done by my friend Chao Wu Chang when we were working together. And actually, he used exactly the same design. You just have seen the sameness design, the same principle that we just discussed together, but to ask whether they can count. So you can see this is the maze he used. This is the entrance. There is an image here at the entrance. Then the bee goes here, enters here, and here there's an image 1 and image 2. And the animals have to match their choice to what they have seen here. That's the same thing we just saw in the previous experiment. The only difference is that, of course, one image gives access to reward, the other not. That's okay. But the only difference is that what he asked to match now is number. So for instance, here, he would show 2 yellow stars. And then when the bees arrive here, they would see 2 blue discs versus 3 blue discs. Okay? So the question is, can they realize that number, quantity, in this case 2, is what gives access to reward? If they can do that, they can count. So in this case, of course, he would change, you see, the shape of the stimuli, the color, because he wants them to get rid of this information. They should focus just on number. So next time the bees would go back, and now at the entrance he would show, say, for instance, 3 yellow stars. And in this case, when they reach this situation, 3 would be the correct choice, if they match based on number. So he did that with many stimuli, situations, and so on. To make a long story short, he was able to show that bees could solve this problem, meaning that they could count. When they saw 2, they went for 2. When they saw 1, they went for 1. When they saw 3, they went for 3, and so on. Where is the limit? The limit is 5. So when you had 5 elements, or 5 was the limit, 5 could go well, but 6 performance collapsed. And that's not bad, actually, because we should consider that other vertebrates can count. It has been shown that primates can count to some extent. Even corvids, crows, can count. There's some very nice research on that. And the limit is around 6 or 7 also. So bees being around 5 is not bad at all. So these experiments show that these little animals can count. A typical question people ask is what this would be useful. And one of the experiments done long ago when I was in Berlin showed that actually counting might be useful in navigation. So by passing a certain number of landmarks and then landing at the right place might be one of the cues they use with other cues for navigation, of course. So, in the last years, we did this experiment based on their sense of number. So knowing that they can count, one of the questions that was actually coming to my mind all the time, do they have spatial numeric associations? What does it mean? It's a complicated term, but just to say something very simple, which I explained you already, which is the idea that we order numbers in space, okay? So we put 1, 2, 3, 4, 5, so from left to right, from small to large numbers, okay? That's what we do. Some people argue, well, but this is based on culture, because we are educated to write or to, and in that way, and that might be the result of culture. Wrong. Because culture, of course, plays a role, and might reinforce this idea or this kind of representation of numbers in space. But you should know that there are experiments with newborns, babies, 2 days old, 3 days old, that show already this kind of ordering. You might ask, how can people do an experiment with a newborn? It's very simple, actually. So they put the newborns, they are lying like that, and they show a screen on top of them. On this screen, they show a number of objects, like, say, 8 objects shown there on the screen. Then the babies are habituating, looking at these images, then they switch off this, and then they show on the left and on the right, say, a smaller quantity. Let's say they have seen 8, and now there is 2 and 2. And of course, the idea is not that the babies count 2 or 8, but they are able to see there is more or there is less, okay? And the experiment shows that when they show, say, a smaller quantity, babies look at the left, just more at the left. And when they show more or larger quantities, babies look at the right. So these and many other experiments show that actually this tendency to align numbers from left to right or smaller to large exists already there. It has biological basis. And so I wanted to know if, given that bees can count, they would have something like that also. This kind of mental number line, it's called, you see, smaller numbers, larger numbers. Do they represent numbers in a way, in a similar way as we do? And I was working with these 2 colleagues, Rosa Rugani, who is a professor in Padova in Italy, and Catherine Thévenot, who is a professor in Lausanne, in Switzerland. She is a specialist, by the way, in children numeric cognition, and she is a specialist in vertebrate cognition. She works with birds and monkeys and so on. And actually, the point is that this was in COVID time, so I was enclosed in my garden in the south of France, and I decided to do the experiments by myself, because what else could I do? It was a good way to spend the COVID time. And then I didn't have access to the lab, unfortunately, but I had a small workshop in my house, and then I took a wine case. You can see it here, actually. Chateau Fontarnais. Actually, I have to say that they are not funding my research, unfortunately. I should show them this image. And I transformed that into a maze. Actually, you can see here, there is the entrance of the maze, and you probably saw the bee coming through and so on. And here, the bees were trained to go in to get the reward here. So the idea is the bees were trained to enter into the maze, you can see here, and they were trained on a certain number. For instance, here you see 3 black squares. So they were trained during 30 visits, consecutive visits to the maze on a number, which was, say, 3. 3 was always there, but it was changing. It could be 3 squares, 3 circles, 3 triangles. The distribution changed, the size, but the number was kept constant. Until the test moment. When they arrived, then we make this image disappear, and then I turn these 2 walls, and then I show, say, 1 and 1. Like the babies, okay? So they were trained on 3, and they see 1 and 1 on the left and on the right. Would they go to 1 on the left or to 1 on the right? In the same way, I could turn the walls and show this time, after being trained to 3, I can show 5 and 5, because I know 5 would work, okay? So that was the experiment. So will the bee prefer one specific site for the 2 identical options? And will this site vary depending on the number trained? So the idea is exactly this. So they were trained, for instance, to 3, and if they see 1 and 1, will they go to 1 on the left, smaller? Or if they see 5 and 5, would they go to 5 on the right, larger? And this is what happens. So they were trained to 3, and here you can see some of the 3s. You see triangles, different dispositions, squares, circles. And then they were tested either with 1 versus 1, or either with 5 versus 5. You see examples of the 5s and the 1s here. And this is what I got. So this is the test in which I presented 1 and 1 after training them to 3. And here I'm showing you choices for the option that was displayed on the left. Okay? So when they see 1 and 1, they prefer 1 on the left already in the first choice. Okay? If you show them, after being trained to 3, 5 and 5, they don't like 5 on the left, which means they like it on the right. Okay? Exactly as the babies. This is another variable. In this case, we counted choices during 30 seconds, but it provides the same kind of reading. As you can see, they prefer 1 on the left, and they prefer 5 on the right. So indeed, like babies, like we do, bees ordered, after being trained on 3, they ordered 1 on the left and 5 on the right. But a typical characteristic of this mental line number we have is that the ordering depends on from where you come. In this case, you were coming from 3. But if you come from another number, you might change the position. In other words, in this experiment, we used 2 groups of bees. One group was training on 1, 1, 1, 1, 1 all day long with different kinds of 1, okay? And another group was trained on 5, 5, 5, 5, and so on, okay? And both were tested on 3. But these guys were coming from 1. So 1, 3 should be on the right. These guys were coming from 5. For these, 3 should be on the left. So is this what happened? And this is what we got. So here we see choices for 3 on the left. Remember, it's exactly the same test for both groups. And here I'm showing you how they preferred 3 on the left. If you came from 1, you didn't like 3 on the left. You like 3 on the right. 1, 3 should be on the right. And that's what they show. If you come from 5, in this case, yes, you like 3 on the left. You come from 5, 3 is on your left. And the same for the variable in which you count the choices during 30 seconds. So this experiment shows this very characteristic of this, very important characteristic of this mental number line. They order numbers from small to large, and actually your ordering depends from where you come. If you come from 5, 3 is on your left. If you come from 1, then 3 is on your right, exactly as in our case. So this shows that not only they can count, but they also represent numbers as we do. And it's another evidence for the convergence between the number sense of bees and other vertebrates. So what is the explanation for this mental number line? So scientists now believe that this is due to the fact that we have lateralized hemispheres. You know, our brain has 2 hemispheres, and they do not work exactly in the same way. Some are, one hemisphere is specialized for some tasks and the other for other tasks. Well, you have to know that bee brains have also hemisphere, 2 sides. You will see that later. And there is also specialization, and that might be the explanation why this capacity is there. So let me move to the third part of the talk, which is actually now going into the brain. I was telling you at the very beginning that it's absolutely great to see all these fantastic capacities there, but for the neuroscientist, he would like to know how this is work. So, and the problem is of this free-flying bee experiment is that you don't have access to the neural mechanism because the bees are flying back and forth. You cannot go into the brain of a flying bee. Unfortunately, it's not possible. So we need to use other protocols to study learning memory and access and having access to the brain simultaneously. And this is why we use actually conditioning or learning experiments in immobilized bees in order to have access to the brain. Bees have to be immobilized for a simple reason. If you have to measure activity of neurons and so on, if the bees move, actually then your signals are lost. I mean, you need to have a full control of the situation. This is why when you go to an imaging machine in the hospital, you are set like that and you don't have to move, because if you move, all is noisy and then you don't get information. So in this case, we will be speaking about olfactory learning, learning about odors, which is actually very important for bees because they learn odorants in nature. And this kind of situation is somehow reproduced in a simplified way in the laboratory, and that's what you can see here. So in this case, what you see is little bees that have been harnessed in small metal tubes. You see them, the head just protruding from the tube. So you can have many bees going through the experiment. And after showing these, after having the bees harnessed, people ask, how do you do that? I mean, it's very simple. You capture them at a feeder, you bring them to the lab, you put them 2 or 3 minutes in a fridge so that they get sleepy, and then you can gently harness them without being stung. Otherwise, it would be too painful, actually. So, and then you put them in face of a machine like this one, which is sending constant clean air to the animal. You see the bees here. And you can actually send the air towards some cartridge in which you have an odorant that you set there. And then the bee will get an odorant stimulation. This odor doesn't mean anything to the bee at the very beginning, so there will be no reaction to this odor. But just after delivering the odor, you will stimulate the antennae with a toothpick soaked in sucrose solution. So what will happen then is you touch the antennae with this sugar water, boom, the bee extends the proboscis, the tongue, okay? So it's a reflex. It's like hitting your leg with a hammer, boom, the leg is extended. It's something like this. But what we are doing now is teaching the bee that every time the odor arrives, there will be sugar reward to touching the antenna, and then she will be allowed to lick the sugar solution. So she will make the association, oh, odor means sugar, okay? So this is just the machine, this is the bee, this is the toothpick, and this is how it works. You will see here a red square showing the odor arriving for the first time. No reaction to the odor, but now toothpick with sugar, pack, extension of the proboscis, which is the tongue. The bee likes that, insists, I want more, it's great, okay? So the bee has hopefully learned that odor means sugar. And now you can see it here. Now we will show just odor, no sugar at all. And you see what happens. Because the bee learned that odor means sugar. So it extends the proboscis to the odor. Okay, it's like Pavlov's dog. I don't know if you know the story of the dog learning that a bell means meat and starting salivating to the bell sound. Well, it's exactly the same, okay? Bees do not salivate, they extend the proboscis. They extend the proboscis not to meat, but to sugar. But here they learn to extend the proboscis to the odor that anticipates the arrival of sugar. So this is very practical because they are incredibly fast in learning these associations. In the movie, I show you one trial, which is one time odor sugar together. Now let's imagine I show you 6 times odor sugar. Imagine that there are 5 minutes between each trial. Okay, so it's like 30 minutes experiment. And then here you're representing the percentage of bees showing proboscis extension response, that's the abbreviation, okay, to the odor. So meaning bees that learn to respond to the odor. I call it odor A, just to call it in one way, and I put a plus just to indicate that there was reward there. And you can see these typically. You see it's actually already after the first trial, half of the bees have learned, and after the second trial, almost the entire population of bees has learned. And so it's incredibly fast. Not only that, now you can take these bees and keep them in an incubator in the dark, feed them regularly, and you don't provide any other odor because you don't want the information they learn to be erased by other kind of things. And you can keep them alive in the incubator for, say, 2 weeks. It's possible, okay? Feeding them regularly. And then you can take them 2 weeks later, bring them back to the lab, place them in the setup, and ask them, hey guys, do you remember what happened here 2 weeks ago? Which odor did you get 2 weeks ago? Was it A, was it B, or was it C? And the typical responses you get in these experiments are like these. Okay? So I used to say it's much better than some of my university students, I would say. But I would love that they have this kind of memory consolidation. But this is why you can see why labs in the world focus on the memory of these animals, because it's so fascinating, it's so great. The other thing is that it's possible to go into the brain of these animals because the cuticle, actually, the external surface is not innervated. So it's just a kind of armory. Then you can cut a window and then go and measure the brain. Then afterwards you can glue it again, and it's possible. We do it regularly, and then the animal is fine. I mean, it's just put it back. And then you can access to this brain and understand what's going on. This is important because we can solve very important questions, as you will see by doing this. The question I would like to address now is whether this brain is plastic. Remember I mentioned that at the very beginning. So your brain changes all the time, believe it or not. Right now, your brain is creating new connections between neurons in order to trying to stabilize or making parallels or something like that based on the new information I'm providing to you. Your brain is changing. You're trying to acquire new information and so on, and new neurons are creating connections to other ones and so on. The point is that some of these connections may be lost tomorrow, and then you forget totally. But in some cases, you might stabilize this information, typically by producing or synthesizing proteins that will stabilize these connections, and this is what will create long-lasting memories. In other words, brains are plastic. They change all the time. This is when people tell you the brain is a computer, you should tell them wrong image, because a computer is made of hardware. It doesn't change all the time. Okay, probably computers in the future might do it. But I mean, the brain is really a plastic machine in our case. So what about these little creatures? So let me now bring you into this brain and to show you the fine connectivity of this brain. Some people might tell, why do I should have to care about this? You will see this is very important to understand the last part of my talk, which is related to pesticides, which is a sensitive issue. I imagine here in the UK too, in France in any case, you know, it's a very sensitive issue. And so we might first need to understand how this little brain is done to see the impact of pesticides on this structure. So I will show you, I mean, you can see here the brain, okay? And you see these very big structures here in red. I mean, we color them red. I mean, they are not red normally. But these big structures here, which occupy more than 30% of the brain, are called the mushroom bodies. Mushroom bodies because with some imagination, you have some mushroom shape here, and you have these two mushroom bodies here. These structures are very interesting because they have been historically associated with memory storage, memory retrieval, and so on. And in fact, it's true because when bees smell, the olfactory receptors are here, and the information goes through the antenna to the brain, and then goes through a series of phases, and then finally reach the mushroom bodies. So the mushroom bodies is the final stage of this olfactory pathway, and this is why people believe this is where the memories are. Not only olfactory pathway, the same for vision. So you see images here, the information goes to this different stage and ends up in the mushroom body. It's like the final place of information processing in the brain. This is why they are considered so important, actually, and associated to memory storage and retrieval. So, and let's have a look at this brain in more detail. So, this is what you see here again. So you have to imagine that the eyes are here. This is one mushroom body, and this is the other mushroom body. Okay, same image, but with more detail. Now what I would like to do, so imagine that the antenna were arriving here, and so on, and so on. And from here, the information goes to the mushroom bodies. Okay, same from the eyes, laterally, and so on. Okay, let me expand this image. I would like to show you this part of the mushroom bodies, which is the entrance region of the mushroom bodies. What does it mean? So others, as I say, come from the antenna, arrive here, and then they go via some neurons to the mushroom bodies, and they do not go everywhere. They go to this specific area, which is the input region of the mushroom bodies. What about vision? Well, same. They arrive to some specific area, which is here. Okay, I'll show you more detail. So let's expand this input area of the mushroom body. In this part, you have arrival of olfactory information coming from here. In this part, you have arrival of visual information come from the optic lobes that were placed laterally. Okay? So now if you look, you see some small structures. Can you see them? Okay, let's make it larger so you can really appreciate them. And you see them. Not only you see them, but I enlarge one of them. So you see a red center and a green surrounding. Well, these colors are artificial. It's me. I colored all the neurons arriving here in red using specific dye, and all neurons building the mushroom bodies in green. I can use these specific dyes to mark these two populations of neurons in different ways. So what are you looking here actually is something absolutely fascinating. Look at the scale. It's 3 microns. It's nothing, okay? It's incredibly tiny. And what you're looking here is the fine connectivity of this little brain. So what is this red spot you see? Actually, imagine that you have a neuron bringing here the information, hey, I'm smelling octanol. Well, this neuron I colored in red. And what you see here is the arrival of the neuron bringing this information. And then this neuron is surrounded by many little neurons which build the mushroom bodies. You can see them here, which say, okay, give me this information. I take it. So every little spot you see here is called a microglomerulus. Okay, the name is probably not important, but the important concept is to have in mind is that every little structure you see is one neuron arriving and saying, hey, I'm bringing you this information, and the green surrounding is the little neurons saying, okay, we take it, and we build this structure, and we take this information. Is it clear? Should I repeat it? Okay, it's fine, great. So this is what you see here. So it's the fine connectivity of this brain region, neurons arriving in red, neurons surrounding and taking the information in green. And they create this microglomeruli. So that's, well, by the way, the green cells I call canyon cells, but that's not important. The important idea is the concept of a microglomerulus that you can see here. Okay, now we know how it's organized. And now let me show you the experiment. Remember, the question is this brain plastic? Okay? And this is an experiment done by one of my students, actually. He had a group of group of bees, but also brains, trained in the same way you saw with odor and sugar, okay? So as you saw it, he provided 5 times odor, 1, 2, 3, 4, 5, associated with sugar, okay? Separated by 10 minutes. Okay, so he knows that bees will learn, as you saw it in the curve and the movie and so on. So he expects that is in group in which sucrose and odor were paired, so put together, there would be learning and memory. And he had a control group, actually, in which he had also 5 times odor, 1, 2, 3, 4, 5, and 5 times sugars, but these were unpaired, disconnected. So there's no way to learn, because these two do not appear together. So they have exactly the same sensory experience, 5 times odors, 5 times sugar, but these guys learn and create memory, these do not. And what he does is he waits. Remember, it lasts for a long time. In this case, he waits for 3 days. He could have waited longer, but that's enough. He waits for 3 days, and then he asks, okay, do you have memory? Do you remember? And you respond to the order that has been learned. Obviously, the answer, you know already, it should be okay, it should be yes. What about this? In this case, there should be no learning or no response because there was no learning here. So let's see the behavior. This is what he got. So remember, first day, he trains. There is a group which gets odor sugar pair, and they learn perfectly, as you already saw, no surprise. And the group which got odor sugar disconnected, there's no learning. There's nothing to learn there. They are not associated. He waited for 3 days. On day 4, he asked, do you have the memory? The pair group, obviously, remember. The group unpair, there's nothing to remember if you didn't learn. But now comes the nice part. So now he takes these 2 groups to the microscope and analyzes the quantity of microglomeruli with the question, or the underlying question, did you change your microglomerular number when you create a memory? Remember, change of connectivity. Did you increase the number of microglomeruli by having formed a memory of another? Is your brain plastic, okay? And he actually did exactly this. So you have some tools in microscopy, confocal microscopy, where you can go, you see the image, but you can go in deep and then count the density of microglomeruli all along your analysis. And this is what he's represented here. He did that in the olfactory region. Remember, this is the part related to odors. And he did also that in the visual region, because he assumed if there are changes, given that it was odor learning, the changes should be just here, not here, because this was not visual learning, it was odor learning. Okay, and this is what he got. So this is the density of microglomeruli. As I say, he counts the microglomeruli and can establish the density. This is the group that did not learn, did not form a memory. It has a basal level of microglomeruli. Look at the group that was able to form a memory. Boom, a significant increase in microglomeruli. You can see here, the little star means that this is statistically robust and significant. So there was an increase in the olfactory region. What happened in the visual region? Well, in the visual region, there are no differences because the change is localized to the odor region because it was an olfactory memory, the one that was created. So, so this experiment shows several things. So first, the big brain is also plastic, like yours. Memory formation, leads to changes in connectivity. You have seen that. New microglomeruli were formed related to this long-term olfactory memory. And the microglomerular density increases, new microglomeruli appear. So if the question would be, where are the memories of these guys? But the answer seems quite simple, in the new microglomeruli, the ones that have been created. This is the place where the memory should be stored. And this is why you see, now I hope you understand why these creatures are fascinating for a neuroscientist. Not only because they are amazing creatures, because all the things you can understand by looking at them. Okay, now let's move to the final part. Now you became experts in microglomeruli, you know what it is, and that's good actually because this will be used for the last part. Okay, I realize that microglomeruli might be very far away from what you're doing, but perhaps this might underline the relevance of the idea. So the question is, what happens when you expose bees to pesticides, and in particular to sublethal doses of pesticides? That's the key word of the story, sublethal. Well, because actually when pharma companies and so on sell pesticides, of course they are not stupid and they are intelligent, and they say, look, if you use the pesticides at this dose, which we have proved to be sublethal, meaning by that it doesn't kill, okay, there will be no problems. And they are right, okay? We cannot say that they are wrong, because they have armies of scientists working with them, or for them actually, and they have done tons of assays to determine which dose is good and doesn't kill individuals like insects in the field. And the question is, of course, they don't kill, fine, but are they doing something else that we do not see, which is hidden? Because they use behavioral tests, like are they returning to the hive, yes or no, or is there mortality and things like that. But they are not going into the brain. Nobody's doing that. So this is what we can do. That's our field. This is what we decided to do. And so the question is, now what happens to these mushroom bodies, to these microglomeruli that you have seen, and you have seen how important they are for memory and so on. What happens when you expose bees to sublethal doses of pesticides, okay? Do they affect connectivity in the brain? And these are experiments that were done by Chinese colleagues. I was telling Roger yesterday that before COVID, I had an invited professorship in China, so I spent a lot of time working with them. These colleagues, Peng and Yan, actually, they did this experiment using the methods we provide to them. So in this case, they treat one-day-old larvae, you can see, with sublethal doses of a neonicotinoid, imidacloprid. Okay? So they just topicate the larvae with these, or they feed them. There might be different ways to deliver, but just when they are 1 day old. So it's very brief. And they maintain the larvae in an incubator until adulthood. And as I say, they don't die. They survive. Sublethal doses, they don't die. They reach adulthood. But what they did when these bees reached the adult stage is they went into the brain and did the counting you have already seen of the microglomerular density. They look at that to see what happened. So essentially, this is what happened. So you had your adult bees actually reaching the adult stage, and these are different images of something you already know. This is the olfactory region of the mushroom body, remember? Okay. Well, they didn't use two colors, red and green. They just used one color because they say, okay, but we can see the microglomeruli already with one color. And indeed, you see them, okay? So they didn't dare to use two colors. One color was enough for them to count the microglomeruli. And this is a control bee. So this is the brain of a control bee, a bee that was not treated with pesticide. You see all these microglomeruli here. This is a bee treated with 1 part per billion. ppb means part per billion. So which means 10 minus 9, okay, of pesticide, which is 0 and add 8 zeros, okay, and then 1. So it's nothing, okay, absolutely nothing. 0, 0, 0, 0, 0, and so on, 1 of pesticide, okay? This is a bee treated with 100 ppb, which is also nothing because it's 0 and 0, 0, 0, 7 zeros and 6 zeros and 1. And this is 500, so it's 5 times this, which is still nothing because it's a really very tiny quantities. And you see already what happened. So 1 ppb, there seem to be some black spots here, but you don't know. But when you have 100 ppbs, you see lots of black areas here. 100 ppb, it becomes almost totally black. Of course, you cannot use this kind of impression to judge what's going on. You need to do the counting, okay? You need to do the counting as you did, as we saw it already. And this is what happens. These are bees that were treated in this way, reach the adult stage. So when you see them, you say, okay, they look normal, okay? Probably, as the companies say, they survived, there's no harm. But let's see what happens in the brain. So these are the control bees. And we set the microglomerular level to 100% to say, okay, that's the level, the reference level, 100%, and see what happens to the other groups. Bees treated with 1 ppb, they have a small decrease, but this is not statistically significant. We cannot say there is a decrease even if it looks like a decrease. However, when we see the bees treated with 100 ppb, boom, look, this is statistically significant, and you have a loss of 40%. 40% of the microglomeruli have been lost. You see, you are here at 60% with respect to the controls. If you have 500 ppbs, the loss is even more dramatic because now you have a loss of 60% of microglomeruli. So what does it mean? It means that, okay, these pesticides do not kill. True, sublethal doses do not kill. But we see a reduction, a significant reduction in the microglomerular density of bees exposed to these sublethal doses of these neonicotinoids. And now you understand. Yeah, it was necessary for me to show you first the microglomeruli and the relationship to learning, memory, and so on, to understand what this means. Now you understand that this loss will have severe consequences for memory, learning, bees getting lost in the field, and so on, and so on, and so on. So the conclusions of this part is that, okay, we saw this significant decrease in microglomeruli, which are key structures for the memory, and this will have dramatic consequences for survival in general. And as I say, these sublethal doses of pesticide, they do not kill immediately, but what they do is to create brain damaged individuals, which amounts to the same thing at the end, okay? So that's the idea. So I like to finish here. It was a kind of selection of topics. We address many other topics, but there's no time. And what I like to show you today, what I try to show you today, is that we have this little brain, tiny brain, which is very attractive because it has a network of areas, neurons, regions that you can study with very sophisticated tools. You have seen some of these today. And these structures, these neurons, produce, of course, some kind of innate behavior, like extending the proboscis or going to the light or something like that, but also plastic behavior. Plastic behavior, it means learning, memory, and we have seen that it's not just simple forms of learning. We have seen actually that this bee brain is neither primitive nor rudimentary. They learn, but they can actually do some kind of, they are able to show some sophisticated forms of learning. We have seen conceptual learning, remember sameness rule, but there are many others we have been shown. We have shown. We have seen counting. We have seen all these capacities are there and many more that there's no time to explain today. And we have seen also that this bee brain is plastic and changes with experience. And at the end, an application of this is to show that pesticides at sublethal doses do not kill, but induce significant brain lesions, which affect survival. So last take-home message is that, well, first of all, at the very beginning, I told you, people ask you, why do you care about bees if you like to understand brain, learning, and memory? The answer should be in the talk. I hope it was pretty obvious why I like to work in these creatures, because I think we learn a lot about ourselves by looking at these little brains. And actually, so this is a brain that teaches a lot about ourselves, and above all, but I mean, I don't need to explain this to you because you share this feeling with me, it's a brain that deserves respect and protection.
Do older bees suffer from memory loss exactly the same as older beekeepers?
Well, one of the things that I can answer is that aging is also a problem for bees. So there are some studies showing that the more aged they are, the less efficient they are in learning and memorizing. So there is even a line of research, that's not our team, that's some team in Norway that looks at aging or the negative effects of aging in a series of performance in bees. That's what I can say. The other thing is that, I think we spoke yesterday during dinner about that. There are bees and bees, okay? When you see all these little insects and they look the same, forget them. Forget that. They are all different. They are in a hive different personalities, okay? And we had a PhD thesis on that. We have a German young woman who got a PhD with us 2 years ago, and that was the topic of her PhD, actually, to realize and conceive a series of tests to show that not all the bees have the same capacities, even if they are all foragers, okay? So some were much better at learning odor problems, others were much better at learning visual problems, others were very good at learning conceptual problems, others were bad at conceptual problems, but good at simple problems. It's like in a classroom, you know, you have all set of possibilities. And our idea is that this is exactly very good, but that allows a colony to tackle all these problems in the environment in a very efficient way by having all this spectrum of possibilities and bees tuned to these or tuned to that. So going back to your question, yes, there are some bees that have better memory than others, but it also depends on which problem we are talking about.
Is there any evidence that bees can pass memories on to other bees?
Memories, not really. Information, yes. There is something that might go into that direction, but it's not exactly that. Of course, bees pass information via the dance that you know. And the dance information actually conveys let's say, information about distance and direction of a profitable food source. Yet there is more than that. I mean, you have to realize that when bees come from a food source, a flower or something like that, they have the odor of the flower impregnated on the body surface. And you probably also know that when bees are dancing, they are followed in the dark, actually, by the followers. And the dancer regularly stops and regurgitates from the crop a little drop of the nectar it has to stimulate the followers. If you capture the followers and put them in the little tubes and then blow the odor that is impregnated on the body surface, then you see they extend the proboscis. Meaning that the dance is not just an innate behavior to report distance and direction, but it gives place also to the possibility of learning and memorizing the odor which is coming from that flower, and then orienting better your search when you go for food. This is something that might go in this direction, but they might not pass memories in that sense.
Absolutely fascinating, and there's hundreds and hundreds of questions.
I'll be around all day long.
Great. I look forward to that. You may not. Neonics. I understand that neonics can stay in the soil.
Absolutely.
For anything up to 10 years.
Absolutely.
So if you've got a colony that starts off, it gets the neonics in, it gets the first generation through, that hits the brain, and so on, the progression. So with your example of how you go 1 particle, 100, 500, have you done any research into how that impacts on them? Because
no, I mean, we have not done these experiments, unfortunately, for 2 reasons. You probably saw that this kind of approach is quite new in our team. And of course, it was actually driven by the necessity of providing some answers to what's going on. So it's quite new, and the experiment you're asking for is an experiment that should go over many years. So we have not done that so far. We would like to. Actually, I'm trying to convince my university to open a professor chair to focus on these particular kind of problems: pesticides, environment, and brains. Let's see if I will be successful or not. and then we will have the space and possibility to address these kind of questions. So I can say no, unfortunately I have no answers to that. My guess is that, of course, the impact will be dramatic, given that you saw it in this very simple experiment. If you have longer exposures and across generation, it should be even worse. But that's just speculation so far.
The neonics in the soil, that's what is it 10 years?
Yeah. I mean, there are some ecology labs, because these are the people addressing this kind of question. Just keep in mind, we are neuroscientists. We don't go to the field to measure these kind of things, but we know some information about that. I know some labs in Montpellier in the south of France with, and some people there who measure that specifically. And if I remember well, what they publish is obviously 10 years, but could be even longer than that. Okay, so, and there was an experiment in which they set the pesticide there and they wait for, I mean, they asked the owner to not use the field, so there was no application after that time for several years, and I was more than 10 years if I remember, so the place was apparently virgin after this application, and then they came back and the pesticide was still there. Again, we have not done work in this direction, but some colleagues working on similar approaches have done work. I don't know if the English word is glyphosate. They did the experiments with that product, glyphosate, and they found also very substantial impairment of learning and memory based on that exposure. But that's not our work. That's another group. Okay. So yes, there seem to be. I mean, we are actually being extremely responsible with our environment. It seems the more we dig into this, the more we realize that, and the more we realize that a change should come somehow in concept, in the way we address these kind of problems. Because one of the things I like you to think is that, okay, pesticides or these products are doing this kind of impairments, this kind of deficits, and so on. But now, if you change your view of bees, okay, most of you are beekeepers, and I'm sure you love bees, okay? But probably you didn't know that they are able of some capacities like counting, like learning conceptual relationship. So it's like looking them with another perspective, which actually should push more respect. And then if we realize that we are throwing all this garbage all around and we are hitting brains that have incredible capacities, then we have to conclude that the change is necessary. I don't know which one, but something that diminishes this impact at least is obviously necessary. We should look for other solutions.
So in a normal honeybee colony, you have about 10, maybe 15 groups of half-sisters.
Exactly.
So does the learning performance of the workers depend on who their father is?
Exactly. So, so you know that a queen is fertilized by many drones in a nuptial flight. So, and this is why you have different patry lines within a colony. And that is related to the PhD thesis I mentioned just before, when I explained you that we found that bees look all the same, but they have at the end different capacities. And some of these capacities relate to the patry line. We have some gene locus or loci, which actually explain why this population might be like this in terms of learning capacities and so on. There are indeed some genetic basis to that. And also for other behaviors like becoming a pollen forager or a nectar forager is also actually based on this genetic diversity and so on and so on. So the answer is yes. That at least we know.
Bearing in mind, we've got a microscopy of a beekeeper's lecture coming up next. I was just wondering what sort of equipment you used to look at the growing microorganisms in the brain,
okay,
and whether or not we could see that with the microscope a beekeeper might have.
Oh, well, these are very sophisticated. We have confocal microscopes, which are very powerful microscopes, which allow you to enter into the depth of the structure without slicing, because otherwise you need to slice the tissue to see the different depth. In this case, these are very complex and powerful microscopes called confocal microscopy, in which you can go in to some depth and then see what's going on. Then we have also some what's called multiphoton microscope, but that's too complicated and too technical. But these are microscopes of last generation, very expensive, by the way. And yeah, I mean, and these are some of the tools we have. I might tell you a little bit more after, but these are not the typical microscopes you might use when you count pollen or something. No, these are huge machines. So a biphoton or multiphoton microscope, just to give you an idea, is the size of this wall, okay? So it's a huge one. You put this sample here, but the whole thing it uses lasers to stimulate the images and so on. So it's a kind of complex setup.
Glyphosates and neonics are having such an impact on the bees' brains. How's it impacting on human beings?
Very good question. I mean, there's a debate on that, but because obviously you cannot do an experiment on humans to see things like that. So there's no way we can address experimentally your question, because we cannot subject humans to controlled treatment with pesticides and then look at the brains. Okay, but at least in France, I don't know, I have no numbers for UK, but I have numbers for France. Agricultural people, I mean, people, farmers and so on, are among the populations which are more subject to Alzheimer's disease, Parkinson, and cancer. So that's for sure. I mean, the numbers are there, and these people are definitely among the high-risk populations. And one obvious conclusion is that might be related to this exposure of products. Again, the problem is that in science, people will answer to you, oh, this is a correlation. So it's a coincident happening of two factors. You're not showing the causal relationship. For that, you need experiments. And unfortunately, I mean, of course, and reasonably, you cannot do them. But at least the correlation seems to be there.